turbo engine running.
Absolute genius.
Get this.
Welcome.
This is the show where we bring you
Science.
What that essentially means is
discoveries,
Advances.
Questions. Research.
technology.
Unbelievable.
Without further ado,
This is The Naked Scientists. Hello, welcome to this week's Naked Scientists. This is the show that brings you the latest breakthroughs in science, technology and medicine with me, Chris Smith. Coming up, how electrically conductive stitches can speed up wound healing. Scientists find the DNA of human victims embedded in the teeth of 2 African lions shot in the 1800s. And the Nobel Prize is explained. Who has won what and what for? Little clue, you don't need AI to help you to solve this one.
From Cambridge University's Institute of Continuing Education, this is The Naked Scientists.
Up first this week, a new study has found that surgical stitches that can pick up an electrical charge can help to speed up the healing of skin wounds. Electrical potentials delivered to the skin by the conductive sutures made new skin cells grow into and heal injuries far more quickly. The findings, published in Nature Communications, are the work of a team of scientists based in China. We asked Steve Jeffrey, who's a consultant plastic surgeon and a specialist in skin repair at Birmingham City University, to take a look at the results for us.
So this paper from China, they've designed a suture which is absorbable, but it's got a metal core of magnesium. They put wounds in rats and they sutured up muscle with this suture, and then they've closed the skin. And then they've discovered that when the rat moves, it moves the suture, and that generates an electric potential. And that acted to improve the healing of the actual wound.
What's the rationale for thinking that? If one looks at a wound, why would some local electricity make a difference?
We know that there is an electrical potential across the wound. If you put a voltmeter from one edge of the skin, the intact skin, and down into the depths of the wound, there is a potential. A long time ago, there was a German scientist called Emil de Bois-Reymond who described the nerve action potential, and he actually cut his finger and did that experiment. He measured the current flowing. So we know current does flow from the edge of the wound to the depth of the wound. And we also know that that current becomes less as you become older, and also if you've got certain comorbidities such as diabetes, etc.
What effect does that current have on the tissue, if any? Can the local skin cells detect that, and do they use it in some way? Presumably if it's accelerating wound healing, perhaps they can.
We think so. We know that when you have an acute wound, the cells at the margin of the wound start to proliferate and migrate, and they lay down extracellular matrix and new blood vessels, and the cells migrate through the wound until they meet each other in the middle. And it is thought that the electrical potential helps guide the healing cells in order to achieve healing.
In this present study then, is the electrical conductor of the suture picking up its electricity from the deeper muscle and conveying that into the superficial layer, the skin, and therefore helping it to heal? Is that the sort of mechanism here?
It is, yes. It's the physical movement of the muscle creating the electrical charge, and then that is being delivered to the wound itself.
How much better was this when you apply this electrical stimulation, admittedly picked up from the underlying muscle, to the wound? How much quicker did they get better? Do you think this is a realistic prospect, using electrical stimulation to heal wounds in this way?
Yeah, definitely. There's some issues with the paper. So, for example, it's talking about wounds in muscle. We don't normally talk about wounds in muscle. We talk about wounds in the skin. But they had significantly better healing. One side experiment they did was in an infected wound model. They were able to show quite remarkable improvements in fighting the infection. So definitely something to look out for the future.
A look to the future of wound care there. That was Stephen Jeffrey commenting on the paper out this week describing electrical sutures for skin wounds. Well, now it's time to take a look at the first of our Nobel Prizes. And our own Will Tingle takes on the prize for physics.
The 2024 Nobel Prize in Physics has been won by 2 scientists
whom the committee say have helped lay the foundation for modern artificial intelligence. They are Princeton University's John Hopfield and the University of Toronto's Geoffrey Hinton. Professor Hinton said that he was flabbergasted to win, but he shouldn't have been. In the course of our lifetimes, his pioneering work on machine learning is likely to change the way that we do almost everything. It has even prompted a huge existential crisis about the power of machines and seen world leaders gather at summits in a bid to address moral panics. In the late 1960s, Hinton read a number of subjects, including natural sciences, the history of art, and philosophy, at the University of Cambridge. This breadth of understanding of the human condition would ultimately lead to his pioneering work on neural interfaces, which helped pave the way for the creation of what we now know as artificial intelligence. His work on deep learning at Google would see the birth of revolutionary tools that powered the creation of systems like ChatGPT, which is now used by hundreds of millions of people and helped him earn the nickname the Godfather of AI. But he did not, as has been widely reported in the wake of the Nobel Prize, resign from Google because he was worried about AI. This is what he told our Titans of Science programme earlier this year.
People have sort of the wrong story. The media loves to make a nice story. And a nice story would have been, I got very upset about the dangers of AI and that's why I left Google. It wasn't really that. I was 75. It was time to retire. I wasn't as good at doing research as I had been. And I wanted to take things easy and watch a lot of Netflix. But I thought I'd take the opportunity just to warn about the dangers of AI. And so I talked to a New York Times journalist and warned about the dangers of AI. And then all hell broke loose. I was very surprised at how big a reaction there was.
Geoffrey Hinton speaking to the Naked Scientists earlier this year. Jeff, of course, is only half of this remarkable story. His co-recipient, John Hopfield, invented a network that can save and recreate patterns. This is, of course, fundamental to the operation of artificial intelligence. Professor Hopfield, who is based at Princeton University, also developed a network that can use incomplete patterns to find the most similar, a bit like using a computer to fill in the gaps. John Hopfield doesn't have a nickname like the Godfather of AI, but we thought we should ask ChatGPT. It suggested the network architect, the synapse sage, or the neuron master. John and Jeff really haven't done badly at all, have they? Congratulations to you both.
Will Tingle, who will be back a bit later on to profile the winners of the chemistry and medicine gongs. The Tsavo maneaters were a pair of large man-eating male lions in the Tsavo region of Kenya, and they were responsible for the deaths of many construction workers on the Kenya-Uganda railway in the late 1890s. The lion pair were in fact said to have killed dozens of people. Now, while the terrors of man-eating lions weren't new in the British public perception, the Tsavo man-eaters became one of the most notorious instances of dangers posed to both Indian and native African workers on that Uganda railway. They were eventually killed by the British officer Lieutenant Colonel John Henry Patterson, and their skulls and skins now sit in a museum in Chicago. Recently, researchers in the city have isolated and sequenced DNA from hairs of the lion's victims, which are lodged inside one of the animal's teeth. Alida de Flaming at the University of Illinois Urbana-Champaign has been telling me what they found.
There were these two lions in Tsavo. It's a region in Kenya. And they're interesting because they've been the topic of many movies, for example, many books, and most famously, they're known for having also preyed on humans. And so those are the two lions that we study in this specific research, and they're currently at the Field Museum in Chicago.
Are they what, taxidermy specimens? Are they just the skeletons? What's there in the museum to see?
So at the moment in the museum, there are actually the two stuffed or rearticulated lions' skins on the one side, and their skulls are actually separate from them. So you can see both the skulls, but also the lion skins that have been stuffed, which is great because that means we had access to the skulls without having to damage any of the skins or actual, rearticulated lions.
So confronted with these two lion remains, how did you then progress this? What did you actually test and what question were you asking?
So we wanted to see if we can identify the prey from the hairs that were compacted in tooth cavities from these two lion skulls, so from their lower jaws. And to do this, we used both microscopy, but we also looked at genetic material that was contained in hair fragments that were in these tooth cavities.
So are you saying then when the lions chowed down, for want of a better phrase, on whatever they were preying on, bits of the hair were ending up embedded in the teeth, and because they don't have toothbrushes, that stuff just stays in there.
That's exactly what happened, right? We don't know exactly how old they were when this happened, but both of them had parts of their lower canines broken off, and this created a cavity or a hole in which hair was compacted as these lions were eating. So the hair would get stuck in there and would kind of compact up.
And what's the preservation of the material like? Can you get meaningful amounts of, I presume you're going for DNA, from the hair that's in that cavity?
Yes, and this is one of the interesting things is a lot of people are aware that to get a really large quantity of DNA, if you've watched any type of CSI show, right, they usually want to have the follicle of the hair shaft of your hair present. But in this case, because we're focusing in on a specific type of DNA that's called mitochondrial DNA, it's a type of DNA that's really well preserved in hairs without follicles. And so that's kind of why we targeted this type of genetic material.
So what, you just grind up the hair and out comes the DNA? You can separate that off?
It's a little bit of a complicated situation where we have to work in a really specialized laboratory, and that's at the University of Illinois, where there's absolutely no external DNA or DNA other than the ones that you're interested in present. And so we take those hairs and we actually dissolve them in a kind of a combination of different chemicals, and that kind of breaks open the cells and releases the DNA, and that's what we use to kind of start out this process of DNA analysis.
And then you read the DNA sequence, which presumably because the mitochondrial DNA of different species is very different, you can begin to pinpoint what sorts of things these animals had been eating.
Exactly. So each individual species has a unique type or sequence of mitochondrial DNA. And so you can use this to identify the different types of prey that these lions were eating.
How do you know, though, that someone didn't contaminate the skull, the tooth, the environment that they were in, and that's what you're picking up? How do you know this genuinely was prey?
So there's kind of multiple lines of evidence that points towards the accuracy of our data. The number one is that we've never analyzed anything like wildebeest or zebra or giraffe in this specialized facility that's at the University of Illinois. The second is when we did this analysis, the type of mitochondrial DNA that we got when we compared it, for example, for the giraffe or zebra to known mitochondrial DNA from across Africa, we find that the lions, the hair DNA matches most closely with other giraffe or zebra that were actually from Kenya and not from the rest of Africa. So that's kind of a good support or validation that the DNA that we're extracting is actually authentically from animals that used to live in Tsavo.
What about the human remains? Could that be mapped to Kenya? Were the locals being picked on, or was it colonists coming in and being picked off?
Yeah, so we are refraining to kind of infer any type of ethnic identity from this really small piece of DNA that we analyzed. We do, however, know that this DNA that we got from the hair had specific damage patterns, so your DNA degrades over time, and it had damage patterns that are characteristic of what you would expect from ancient DNA or DNA that's really old and have degraded over time.
We've known that we could do this for mitochondrial DNA in sort of related ways for some time. What is new about this study, and what does it add?
So I would say two things. First is methodologically, this is interesting because where folks have used ancient hairs to do any type of research, usually you know what type of animal you're actually sequencing or getting DNA from. A good example is people have analyzed hair from Siberian mammoths. In our case, we didn't know what the hairs were from, and so we kind of take a backwards approach and say, we have a bunch of DNA, can we identify the actual animals that they're from? So that's a type of novel way of interpreting paleogenomic data or ancient DNA data from animals or from hairs. The other kind of interesting aspect of this project is it also gives us kind of insight into the historical ecology or interactions that these lions had with their environments. For example, the presence of wildebeest in the area, which is, until we did the study, folks hadn't reported wildebeest, at least from that specific area in Tsavo. So it does give us some insights that we didn't have before.
So research you can really get your teeth into. That was Alida de Vlaming, and that study's just come out in the journal Cell Biology.
The Naked Scientists podcast is produced in association with Spitfire, cost-effective voice, internet and IP engineering services for UK businesses. Find out how Spitfire can empower your company at spitfire.co.uk.
Music in the programme is sponsored by Epidemic Sound. Perfect music for audio and video productions.
And this is the Naked Scientists podcast with me, Chris Smith. Still to come, how large webs of DNA can help to protect our kidneys from infection. Now though, Will Tingle's back with more Nobel Prizes. A trio have scooped the next award, although, as we'll hear, it hasn't been shared out quite equally.
The 2024 Nobel Prize in Chemistry has been won by two researchers from the AI company DeepMind. Its co-founder, Sir Demis Hassabis, and researcher scientist John Jumper. They share the award with the University of Washington's David Baker. All three have worked in one way or another on the structure of proteins, the three-dimensional molecules that make all of life possible. Baker won for his work on creating entirely new synthetic kinds of proteins not found in nature, but which can operate as drugs, vaccines, and sensor molecules. Meanwhile, the DeepMind duo are recognised for their pioneering work cracking the hard problem in biochemistry, predicting the structures of proteins from the sequence of the gene that encodes them or the amino acids that compose them. It's a key nut to crack because knowing the shape of a protein molecule is critical to understanding its behaviour and contribution to life processes, role in health and disease, and potential as a drug or therapeutic target. Hassabis and Jumper achieved their breakthrough by harnessing the artificial intelligence muscle they'd developed at DeepMind. Their tool, called AlphaFold 2, achieved what scientists had tried and failed to do for decades. According to the Nobel Prize Committee, the work has revolutionised chemistry and led to the mapping of the structure of around 200 million important protein structures globally. Interestingly enough, the Nobel Prize for Chemistry hasn't been split evenly. Hassabis and Jumper will share half of it, and the other half goes to Baker. I'm not sure they'll mind though. Hassabis has come a long way since his days of writing computer games. Before joining Queen's College, Cambridge as a student, he took a gap year and wrote the blockbuster theme park simulation game. This is what he told Chris Smith on this very programme all the way back in 2009.
Although I was in the games industry for a long while, underlying all of that, all the games I've been involved with designing and programming actually composed a lot of AI in those games. And most of the games were big strategy.
Artificial intelligence. For the non-initiated like me.
That's right. And all the games I wrote, like Theme Park and Republic and Evil Genius, these games, they all involve simulations. And most of them involved hundreds of little computer people coming and getting involved with the game environment. And then most of the games were, like, for example, Theme Park, were involved you manipulating that environment and seeing how these autonomous agents reacted to what you were doing. Those are the kinds of games I found fun to play, and they were the kinds of games I found fun to create. But so underlying all of this was my passion. My main passion is actually in artificial intelligence, and related to that, as soon as you start thinking about what artificial intelligence is, then you start thinking about how is it that the mind achieves these end goals.
He wasn't wrong, was he? It goes to show that you can indulge your interests and have a successful career. It might even help you win a Nobel Prize, or at least a share of it.
Will Tingle. Researchers at the University of Cambridge have found that webs of DNA can help to protect the kidneys from bacteria that cause common urine infections. These webs, which are called neutrophil extracellular traps, or NETs for short, are produced by white blood cells called neutrophils, which are attracted to the kidneys and then persuaded to spew out their DNA into the urine stream, where it in fact acts as a defence against microbes trying to gain a toehold. Andrew Stewart is part of the Cambridge University team that conducted the study, and I went to see him at Addenbrooke's Hospital.
The question we were asking had nothing to do with what this project turned out to be. So we were trying to work out what in urine we could see to diagnose kidney disease. And mysteriously, we found there was lots of DNA in people's urine, healthy people.
Whose DNA?
It was the patient's DNA, but in fragments within their urine. There's a phenomenon known where cells can eject their DNA as a defense mechanism, so we speculated that this might have something to do with it.
So it's cells from somewhere in the urine producing system are basically chucking DNA out into the urine.
Yeah, exactly. So in order to work out what cell it is, because all cells contain the same DNA, we looked at the proteins on them. And these proteins suggested it was coming from a white cell, and a very important white cell called a neutrophil that fights bacteria, which causes urinary tract infections.
But you said these are healthy people.
They are. So this was really surprising that healthy people seem to be undergoing some sort of chronic defense mechanism. And given this is in the urine, it's logical that maybe urinary tract infection was important here.
Is this some kind of defence then? They're healthy, they haven't got an infection, and this is why?
Absolutely. So we then moved to look at mice, because we can give mice urinary tract infections. So you get a very small catheter, and you put it into the mouse's bladder, and you put some bacteria in, and the mice get a urinary tract infection. So when we did this, we then looked for these nets in the urine that we were seeing in healthy humans, and we could see the same thing.
But the mice have got an infection, unlike the humans. So can you stop the mice having these nets and see if they're more prone to infection?
Absolutely. So there's a drug that isn't used for anything clinically at the moment in the hospital, but it stops these nets from forming. So we gave some mice this drug versus some mice who didn't have the drug, and we found that the mice who had the drug were much sicker. So in the absence of these nets, they got infection that went from their bladders, which is usually mild, up into their kidneys, which in humans can be life-threatening.
Where are the white blood cells that are making these defensive nets hanging out then? Have you now gone and profiled through the urinary tract to see where they're starting out from?
We don't really know the answer to it, but there's good evidence from the lab that the kidney has chemicals that pull these white cells towards it in normal health, in normal healthy humans. And this is then probably increased in infections.
A million questions now running through my mind because, as you say, UTI is a big, common clinical problem. If these are so good at fending off infection, if we turn them up, can we cut down the rate of infection? And are people who are more prone to infection deficient in these?
We actually did look at this question. So we looked at some genetic information, and we found that there was an association between the enzyme that we were inhibiting to prevent these nets from forming and infections, but also autoimmune diseases. And essentially, it's a trade-off of one versus the other. So if you have increased risk of infection, you can reduce your autoimmune disease and vice versa. So if you turn them up in your genetics, you're probably actually going to get more autoimmune disease and less infection and vice versa.
When people go to the doctor, usually women, and they say, I've got some symptoms of a urine infection, the first thing we do is we reach for these sticks that you dip in the urine. One of the things we look for is evidence of leukocytes, white blood cells. So are we finding these things? And it's a good thing if they're in there. It doesn't mean they've got an infection.
That's a really interesting question. And we thought the same thing. Well, what does this urine dipstick test actually tell us, and what does it mean? This test is 70 years old now. So we actually thought, let's have a look at that test and see what it was doing. So we took some neutrophils from blood from a healthy person and did this several times and found that actually lots of neutrophils did not activate this test. So that's kind of contrary to what we've thought for 70 years. So then we took those neutrophils, the same neutrophils, and triggered them to do the net formation. And they activated the test straight away. So this kind of demonstrates that actually what this test that we've been doing and using on millions and millions of people all the time, probably the commonest test we do after some simple blood test, is actually detecting the DNA being ejected by these cells, not the cells themselves.
It's detecting the defence, not the infection it's designed to prevent, so it could be misleading us.
Exactly. So some types of infection are good at triggering net formation and some are bad. And there are many reasons why nets may form independent of infection. And therefore, this test is actually much more complicated. And this is probably why this test is so difficult to interpret in our population.
So where next then?
So I think we've disrupted them, and we now need to understand what drives their formation and what doesn't. Because one interesting characteristic of this cell is it's quite unstable. It likes to do this. But a lot of the cells weren't doing it. So working out what makes them do it, what makes them not do it, how that changes from person to person, and whether that changes in disease, I think are really interesting avenues.
Andrew Stewart there, and that work just came out in Science Translational Medicine. And now it's time for our final instalment of Nobel Prize winners, and it is the turn of US duo Victor Ambros and Gary Ruvkun, who've scooped the gong for medicine. And here's Will Tingle to tell us all about it.
The 2024 Nobel Prize in Physiology or Medicine has been awarded to the University of Massachusetts' Victor Ambros and Harvard's Gary Ruvkun. for the discovery of a key process that controls which genes are turned on or off in different cells in our bodies. This matters because there are more than 200 different types of cells doing very different jobs all around the body, yet they all carry a complete and nearly identical copy of our individual DNA code. So whether it's a liver cell breaking down the booze consumed by a Nobel Prize winner after they've popped the champagne cork, or a cone cell in the retina enabling us to see, these different cells are all dipping into the same DNA recipe book for the instructions that make them work. The difference is that the liver cell will be reading and making a different repertoire of recipes than the retinal cell. So how is that achieved? Before Ambros and Ruvkun came along, the explanation centered mostly on signals called transcription factors. These are small molecules made inside cells that can lock onto DNA and physically turn groups of genes on and off. But what Ambros and Ruvkun showed using microscopic worms called C. elegans is that pieces of genetic material also do this job. Cells transcribe genes from DNA into a temporary intermediary called mRNA. This is a bit like jotting down a copy of the recipe on a piece of paper to avoid having to take the entire Jamie Oliver tome into the kitchen. It's this mRNA copy that's shuttled out to the part of the cell that turns recipes into reality and effectively cooks the cake prescribed in the DNA. But the mRNA can be ambushed en route, Ambros and Ruvkun found, by other short pieces of genetic material called microRNAs. These resemble the mRNA made from working genes, except that many of them are the genetic mirror images of parts of those genes, and if they meet their mRNA mirror image, they can bind on, triggering the cell to rip up the recipe and blocking the expression of the gene. Initially, the scientific community was skeptical and regarded the finding as a peculiar foible of the worms the scientists had been studying. But in the years since, the phenomenon has been confirmed to be operating across the tree of life, at work in even bacteria, through to our own cells. It's arguably one of the most important ways in which genes are regulated, and also offers us a new way to get a handle on important diseases. Because this system is disrupted by conditions like cancer, which can make cells grow in ways they shouldn't, and infections like the herpes simplex virus exploit these mechanisms to cause repeated painful cold sores. But thanks to this system, for the majority of the time, the 37 trillion cells in an adult human know their places and roles. And that liver cell knows that it needs to turn on the alcohol dehydrogenase gene. Very important when you've just won the Nobel Prize and are celebrating with a well-earned glass or two of bubbly.
Will Tingle there now, thanks to him for an excellent roundup on this year's Nobel Prize gongs. Well, now it's time for Question of the Week, and James Titco's been looking at this spine tingler sent in by listener Jim.
Sometimes when I pinch near my knee, I will feel a tingling or tickling sensation behind one of my shoulder blades. What's up with that?
Thanks for your question, Jim. You've touched on something that's actually quite common there. In the nervous system, nerves receive activation at the level of our skin. They send their signal all the way to the spinal cord, where they're processed, and the signals then go on an ascending pathway to the brain to be interpreted. That critical point in the middle there, at the spinal cord, is probably the source of this confusing phenomenon. Here's Mark Hoon, Senior Investigator of the Neuroscience of Sensation at the National Institutes of Health in the US.
Along the spinal cord, there's something called a dermosome, which is just a fancy word to say that there's different units all the way along the spinal cord, and each of those correspond to a grouping of nerves that go to a specific part of the body. It's a very defined area of the body that goes to a single part of the spinal cord, and that unit of information then goes up to the brain, where there's this discrimination of location.
So what's happening in Jim's case, where the stimulus is occurring in one part of the body, but he's feeling the sensation in another? Is it literally a case of the wires or the nerve pathways getting crossed?
In some ways, yes, you could refer to it as that. I mean, it's basically that there isn't a separation of some nerves coming into very precise locations, and that's called referred pain. And a very good example of referred pain is people who have pain that's actually the result of something going wrong in their heart, so like a heart attack, will often not feel the pain close to where their heart is, but will actually feel the pain in the shoulder, the neck, and the back. And very often, if you have a shoulder pain, for instance, the doctors will be quite alarmed and will want to start a test for you to see that you haven't got a heart problem.
And this is why, as you say, there are parts of the body that are more commonly mixed up with each other. The dermatomes for the parts of the body they relate to happen to sit next to each other in the spinal column.
Yes, that's exactly how it is.
Jim's feeling he referred to is more of a tingling than a pain. Some people might have an itch. Do we have a handle on the distinction between them?
A tingling sensation is better described as paresthesia, and that is something that most people will recognize as something that happens, for instance, when they sleep on their arm. You're actually cutting the blood flow, and the nerves are actually affected by that blockage. When you wake up with your tingling arm, it's when the blood flow starts to go back to the nerves, and they all start to fire. And so you get activation of nerves that produce the sensation of pain, itch, and also touch neurons. So it's a funny little activation that causes a tingling sensation.
I hope that's answered your question, Jim, and thanks very much for sending it in. Next time, given it's spooky season, we thought we'd answer this question from listener David. When a spider makes a web, is this passed on through its genes, or does the spider have to learn how to make one?
And if you've got an answer, or in fact a new question for us, do get in touch. You can write to chris@thenakedscientist.com. You can also get involved in the discussion around these questions over on our website at nakedscientist.com/forum. And that's all we have time for this week. But do be sure to tune in on Tuesday when we'll be asking, is a vegan diet a healthy diet? The Naked Scientist comes to you from the University of Cambridge's Institute of Continuing Education. It is supported by Rolls-Royce. I'm Chris Smith. Thank you for listening, and until next time, goodbye.